In this video lecture, we will discuss about the genetic engineering. What is genetic engineering? Introduction about genetic engineering, history about genetic engineering, what are terms included in the genetic engineering, and the process of genetic engineering. So, first of all, what is history? What is been from where the genetic engineering is been developed or been came into the existence? Who was the father of genetic engineering? The father of genetic engineering was the Paul Berg. Here the Paul Berg was known as the father of genetic engineering. He was the first to develop the recombinant DNA technology, means RDT technology, recombinant DNA technology, from where this genetic engineering or the era of genetic engineering began. The first genetically modified animal was the mouse, created in 1973 by the Rudolf Janisch. In 1993, an antibiotic resistant gene was inserted in the tobacco plant, leading to the first genetically modified plant. In 1978, the technology was commercialized with the production of insulin. In this year, the insulin was been developed or came into the existence. While in 1994, the first genetically modified food, tomato, was made. This all are the first animal, plant, or pharmaceutical product or crop. These are the first made genetically engineering animal, plant, or crop. What is definitions? What is genetic engineering? The change in the genetic makeup of the living cells by inserting the desired gene through a vector in is called genetic engineering. The what is definition? The change in genetic makeup. What is genetic makeup? The whole DNA, which is known as genetic makeup, the whole makeup or the design or contents of DNA will be changed by inserting the desired gene through a vector into the host organism or in the organism. This process is known as genetic engineering. What are terms included in this genetic engineering? Gene. What is gene? Gene is a small part of a DNA that encodes for the specific protein. Gene is a small part or a small segment of a DNA which codes or which encodes for a specific protein. What is recombinant DNA? The DNA formed by joining DNA segment of two different organisms. Recombinant DNA means the DNA formed by joining DNA segment of two different organisms is known as recombinant DNA. What is recombinant DNA technology? The technique in which the gene of interest is transferred to the host is known as recombinant DNA technology. The technique in which the recombinant DNA is being developed is known as recombinant DNA technology. What is genetically modified organisms, or else we can say GMO in short? Genetically modified organism, the organisms whose genetic makeup is altered or changed by using rDNA technology or recombinant DNA technology. What are the tools? Which are the tools used in this genetic engineering? First of all, gene of interest. The which gene is been in the gene of interest means the gene we required in our organisms to be produced. The vector may be plasmid or the bacteriophage. Third one is restriction enzymes. Fourth one are the ligases. Vector may be plasmid or may be bacteriophage. What are restriction enzymes? Restriction enzymes are the enzymes which are used for the cutting of the DNA molecules. And while ligases, what are ligases? Ligases are used for joining the DNA molecule. Further, we will discuss about all this. First of all, restriction enzymes. These enzymes which cleaves the DNA from the particular system, meaning the enzymes which cleaves. What is the meaning of cleave? The cleave means cutting the DNA from the particular sequence. The sequence from where it cleaves the DNA is called as recognition site. The specific DNA molecule or the segment is being cutted or been cleaved. Those site is known as recognition site, and recognition site can be 4 to 8 base pair long. It breaks the nucleotide bond of the base pair. How this enzyme works? It breaks the nucleotide bond of the base pair. There are different types of restriction enzymes. There are three main types of restriction enzymes, that are type 1 restriction enzymes. They are made up of three non-identical subunits which requires ATP and Mg2 plus for the activations. They cleave the DNA about 1000 base pair away from its recognition sites. What is in the type 1? They are made up of three recognition non-identical subunits. They require ATP and Mg2 plus for the activation. They cleave the DNA 1000 base pair away from its recognition site. This is all about the type 1 restriction enzyme. About the type 2 restriction enzymes, they only require Mg2 plus ion for the activation. They are made up of two identical subunits, cleaves the DNA from the recognition sites. More than 300 enzymes are been discovered in the type 2 recognition site restriction enzymes. While in type 3, they cleave 26 base pair away from its recognition sites. In type 3, they doesn't require any of the ions or any of the ATPs to be activated. What second one is gene of interest. The gene of interest is a DNA fragment. It is stored in a gene library. There are two types of gene library: genomic library or cDNA library. cDNA library means chromosomal DNA library. In genomic library, what is genomic library? A collection of clones that contains all the DNA segments of the genome of an organism is known as genomic library. While cDNA library, what is cDNA library? cDNA library is an chromosomal DNA library. It is a collection of clones. Clones means segments, different segments. Clones of each of which carries cDNA of an organisms. Which carries means which have the chromosomal DNA of an organism. The third one is vector. Vector means vehicle, which work as a vehicle. A vector is a DNA molecule that has ability to replicate autonomously, means they can replicate itself or does not require any of the helper. It in a host cell into which the DNA fragment is been cloned. It is an extra chromosomal DNA or genomic or DNA. It is a self-replicating vector. For example, who can work as a vector, such as plasmid, phage, in phage, which phage? Lambda phage, phage M31, cosmid, phasmid, BAC means bacterial artificial chromosome. Another one is YAC, East artificial chromosome. This all can work as an vector in the rDNA technology. What are the properties of a good vector? They should have origin of replication, should have specific origin of replication, should be less than 10 kb in the size, easy to isolate or to get extracted and get purified, easily introduced into the host cell, should contain unique target site from many restriction enzymes. Each restriction enzyme or each vector should have different site or unique site for different restriction enzymes. The fourth one is the ligases. What is the work of the ligases enzyme? To join the two different DNA segments. The DNA ligase enzyme, which acts as a glue sticking foreign DNA to DNA of the cloning matter, which works as a joining material or joining for foreign DNA to join with the host DNA, will work only if the DNA to DNA. These ligases will work only if DNA will bind to the DNA. What is the process of the genetic engineering? There are mainly 5 steps involved in this process. First one is isolation, to isolate the plasmid, or at the one side we will isolate the plasmid, at the another side we will isolate the gene of interest. Second one is cutting by with the help of restriction enzymes. Insertion or ligation or joining the two DNA molecules. Transformation, and last one is expression. Here we will take the DNA or the host cell from which we will isolate the plasmid, and we will cut the segment with the help of restriction enzyme. At the other end, we will cut or cleave or isolate our gene of interest here, which works as a foreign DNA. Here the gene of interest we will cut or cleave with the help of restriction enzymes. By cleaving both the plasmid and gene of interest, we will ligate or we will join with the help of DNA ligase enzyme, which will ligate or will join the two DNA molecules. Further, this plasmid through which the gene of interest is been cleaved or been ligated will be transformed or will be inserted into the bacterial cell or any host cell, through which this plasmid which contains the gene of interest will be inserted into the bacterial cell, and they will transform and been replicated into the many different forms. What are the applications of genetic engineering? It is used in human medical treatment, pharmaceuticals, or in industries. In human cloning, it is used to treat the genetic disorders and cancer, help in supplying new body parts. This what it is also used in the medical treatment. How it is used in medical treatment? For the production of insulin, in gene therapy, or human growth and growth hormones from the pituitary gland. It has been also used in the pharmaceutical industries to produce the large number of low cost or high quality new drugs, diagnostic reagents such as human insulin, urokinase, interleukin, erythropoietin. This process is also used in the industries. To using with the help of microorganism, they will produce all sort of molecules, optimizing molecules by mutagenesis means there will be mutation. They will create a mutation in the DNA or in the gene, which will produce by the genetically modified organisms or bacteria. This is all about the process, introductions and applications of the genetic engineering.